I'm going through exam practice question focusing on gene expression, in particular transcription factors, how it affects gene expression, and also RNAi, the interfering RNA. Question number 1. Testosterone is a steroid hormone and it belongs to a group of male sex hormones called androgen. Testosterone hormone are hydrophobic. Explain why steroid hormone can rapidly enter a cell by passing through its cell surface membrane. So when I read this question, first of all, I'm going to focus on these key words. So testosterone, if you know this hormone, if you don't know, it does not really matter. Keyword is a steroid hormone. So steroid hormone, so fatty is made from fat, made from lipid. So just like your estrogen, it's a steroid hormone. And all the other word is a male sex hormone, belongs to or called androgens. These things are extra information which is not very useful here. The second thing is trying to emphasize here is a hydrophobic. So again, so talking about the lipid, so giving you a hint that this is just like estrogen that you've covered in your spec. So it's hydrophobic and a steroid hormone. Explain why steroid hormone can rapidly enter a cell by passing through its cell surface membrane. So this builds on your previous knowledge, and what is that? Is only on estrogen. Estrogen, as you know, that is a steroid hormone, and it just goes through the cell membrane, diffuses in, because it's lipid soluble, and they want you to sort of build that on. So if they ask you in the exam any hormone, any of them that is steroid hormone, that is lipid soluble, that is hydrophobic, it does not require a receptor. It can straight away go into a cell through a cell surface membrane. So the answer would be, let's check out the answer. Mark scheme says that it is lipid soluble, number 1. Lipid soluble, and number 2, because of that it diffuses through the phospholipid bilayer. It does not require a receptor or a channel or a carrier protein. Question number 2. In the cytoplasm, testosterone binds to a specific androgen receptor. An AR is a shorthand for the androgen receptor, is a protein. Suggest and explain why testosterone binds to a specific AR. So in here, a bit tricky. So in the cytoplasm, this hormone has entered from outside into the cytoplasm, and it binds to a specific androgen receptor. So the word specific is very, very important, because we know that in A-level is everything really mostly is about shapes and having a complementary shape for each other to bind. So testosterone is binding, having a specific shape, so it's complementary to androgen receptor. And androgen receptor is my first time hearing this, and so therefore I'm thinking what that is. It's a protein. Explain and suggest and explain why this hormone binds to the specific AR. All they're asking you is just to use the concept of similar things, similar shapes binding to each other. So what could you say? So look at the mark scheme. Mark scheme says that it has a specific tertiary structure. It's all about structures, and you have to mention the word tertiary structure because of the fact that it's a protein. So it has a specific tertiary structure and which are complementary to each other. Question B-C, this sort of builds on, it's a 2-mark question. And then here, the binding of testosterone to a AR changes the shape of AR. So this hormone binds to this protein, and the shape of that protein changes. And you're thinking, where have we seen this in module 8? And it should come to your mind that we are talking about transcription factors. And remember, we said about transcription factors is a general name. There are so many different types of transcription factors. In here, we are dealing with AR. AR is a transcription factor. When the hormone testosterone binds to it, it changes shape. And then to reinforce and just to make us even more convinced that this is a transcription factor, it now moves from the cytoplasm into the nucleus and then stimulates gene expression. Now there should be no doubt from the A-level spec that what this molecule is talking about. This is about a transcription factor. So just how this AR, in other words, transcription factor, could stimulate gene expression. And we talked about transcription factors. They are either repressors or activators. So the word stimulate would hint that this is a activator. So Mark Gims says that AR transcription factor is first of all, you're mentioning what it is a transcription factor. It binds to the promoter region. Okay, I would be more keen to use the word safer to use the word promoter region. So it binds to the region at the start of the DNA, promoter region, and this is where it's going to stimulate RNA polymerase and initiate transcription. Let's have a go at this question. The gene that codes for AR has a variable number of CAG repeats. So the gene that codes for this transcription factor has CAG repeating so many times. Now, some studies have shown that association between the number of CAG repeats and the risk of developing prostate cancer. So some studies have shown that if you have these repeats, okay, you can have if you have certain repeats of them, then there's a link to prostate cancer and the risk there. So this is the study here. So it shows you 16 times CAG is being repeated, 17 times is being repeated, 18 times. Now they are saying that this repetition is linked with cancer, with prostate cancer. They've done a stat test to tell us which number repetition is the most likely linked to prostate cancer. So you will never be able to answer this question if you don't understand probability. So just to remind you, the p-value 0.05 is 5%, and this is what we generally use for A-level, 5%. So if our p-value is less than 0.05, what does that mean? It means it's less than 5%. So what does that mean? It means that there is less than 5% probability that our data is due to chance, less than 5%. So we want our data to be under 0.05 to be convinced that we can trust, and then there is a significant difference. Looking at these values, obviously they will not give you percentages, too easy. They're giving you decimals. So our reference point was 0.05 is 5%. So 0.02, that's how much percent is that? 2%. That means the under 16 or less, having that repeat, that data being accurate, there's less than 5% probability is due to chance. So I quite like that data here. So this is a trustworthy data. All the other ones, 0.3, 30. Oh, this is 30, is really, really high. 0.7, 7%, 9%, 6%. All of the other ones, they are above. They are more. They are 5% or more. They are more than 5%. So you have to understand this probability to. So what can you conclude from the data? From the data, I can see that only the first column, only 16 or less, is less than 0.05. Everything else is more than that. So have a pause and see what can you write for 3 marks before I show the mark scheme. Okay, so mark schemes will tell you this 3-mark question. So you can say with 16 or less is significant. And why is it significant? It is significant, let's see if they mention it, is because, number 2, is significant because there is less than 5% probability of being due to chance. And then you can mention the other ones, the 17 or more, because we said that 17 or more, all of them, they are not significant. Why they're not significant? Because there is more than 5% probability of it being due to chance. And remember, we mentioned for our A level we are using 0.055% most of the time. Okay, so let's go into our next one. HIV is a virus. Particles, so the HIV virus have a specific protein on its surface. So the key word is the word specific protein. That means HIV virus has a protein, which we already should know from our biology classes, on the surface. I know that protein on the surface sticking out will bind to something, right? So this protein binds to a receptor on the plasma membrane of a human cell. So on a human cell, they haven't specified, but we know that it is T helper cell. So the human cell has a receptor outside, and the HIV binds to it. What binds from the HIV? The protein. And that binding allows the HIV to enter the cell. This HIV protein is found on the surface of human cell after they have become infected with HIV. I put that in blue highlighted because students will read all of them. But when it comes to that, they probably will read it quite fast and will not link to this. So have a look at this question. What is it saying to you? It's showing you what? It's showing you a APC cell, antigen presenting cell. So that human cell, they haven't specified, but it's likely to be a T helper cell. It has taken in the virus, and then it presents the virus antigen, virus protein on its own surface. So what has it become? Antigen presenting cell from our immunology module. Scientists have made siRNA. So these are interfering RNA to inhibit gene expression of a specific HIV gene inside a human cell. So how will that work? So we know that, so this requires our knowledge of siRNA and also our A-level knowledge how HIV infects the human body. They attach this siRNA to a carrier molecule. So maybe some of you might think, what is a carrier molecule, right? Carrier molecule. So generally, if you want to give any medication, anything that is outside the body, any medication, and you want it to go to a specific cell, you just cannot give that medication without having a carrier transport mechanism to be directed to a particular protein or to a particular cell. So this siRNA has to be attached to a carrier molecule. Why? Because we want this siRNA is targeted. It's going to specific cells, not any cells, not your heart cell, liver cell, or healthy cell. It's going to infected cells, not the normal cell. So it has to be targeted and guided. So it's attached to a carrier molecule. The carrier molecule takes it to those infected cells. The flowchart shows what happened when a carrier molecule reaches the human cell with HIV. So this is a quite a nice text, but it sort of draws in the different disciplines. So you have to have a good understanding of HIV. You have to understand proteins and receptors, they love to stick to each other. You have to understand how HIV infects the human body, and siRNA and carrier molecule. This is the flowchart. So once I have sort of read this and analyzed this, I have to understand this flowchart. So carrier with siRNA. So this is my siRNA with the carrier molecule. Now it's inside the body. Once it's inside the body, it binds to the HIV protein on the plasma membrane of infected human cell. So I can just summarize that by saying this carrier molecule with this siRNA binds to APC cell, antigen presenting cell of the HIV. And then what happens is that it enters the cell, and then siRNA, it binds to the mRNA of the infected cell. Which mRNA? The cell has so many mRNA. mRNA, HIV mRNA. The HIV that was made into a DNA by reverse transcriptase and then inserted into the human DNA and then became an mRNA in the process of becoming translation into a virus. Is the mRNA. This is the virus. HIV mRNA is just about to be translated into a virus. So siRNA is going to bind to the mRNA transcribed from HIV gene, and mRNA from HIV gene is not translated, and that should tell you why, because siRNA has bound to the mRNA and it sort of cut them into small fragments and small pieces, and then they are degraded. So we know RNA is obviously our if you cover this topic, you will know the siRNA is a double-stranded, then it becomes a single strand, and one of the single strand complementary binds to the mRNA. So having this knowledge, going over, understanding it really, really well, then you should have a go at these questions, not rushing into it, so therefore you don't misunderstand anything. When siRNA binds to the mRNA, so it's complementary, okay, single strand there, RNA, there's no DNA here, name the complementary base pairing that holds the siRNA and the mRNA. So you are doing complementary base pairing for RNA. So have a go, what do you think? So the answer is cytosine with guanine and adenine with uracil. siRNA would not affect gene expression in cell infected with HIV. Sorry, siRNA would only affect gene expression in cells with HIV. Two reasons. So this siRNA with a carrier molecule, okay, has gone to those APC cells, right? So an APC cell, as we know, that it's got this particular it's showing those proteins, right? These antigens outside. Now, why is it that it will not affect other cells? Why is it very targeted? Why do you think it's very targeted? So this is a 4-mark question. Why siRNA only affects gene expression in cells with HIV, not other cells? So this is twofold. One fold is you have to know specifically about siRNA, and the other is a generalized concept in biology. So the first one, well, there's many ways you can say. There's two reasons. The first one I would say is going to be like a generalized one. siRNA, remember, if you go backward, what did I say? That it has a carrier molecule, okay? And the carrier molecule, what does it do? It binds to the carrier molecule. So it's here. The carrier molecule binds to the carrier molecule binds to the HIV protein. So remember, I told you about the complementary shaped concept. You use the same concept here as well. It's always being used in biology. They love to use the complementary similar shape. So the carrier protein has a certain shape, and the protein sticking out on that cell, APC cell, which is the HIV protein, the antigen, is also complementary to the carrier molecule. So other cells will not have that protein, so you won't bind to it. So that's the first generalized point you can say, that only infected cells have this HIV protein on its surface. So the carrier only attaches to those cells, to that particular protein. No other protein will it bind to. That's the first one. And that you can answer without having any knowledge of siRNA, how it works. The second part is you need to understand your siRNA. We know the siRNA, what is it? It is going to target. It's going to target what? This HIV the mRNA from the HIV gene. So the siRNA has to be complementary. The base sequence can't be random. It has to be complementary. So siRNA has a complementary or a specific sequence to the mRNA. And if you go to other cells, let's say you go to your heart cell, and this siRNA binds to your heart cell, will it do any good? Let's say it enters somehow it enters the cell, but where will it bind to? It has to bind to a mRNA of HIV gene, and you won't find it in a heart cell, so it will not work there. So that's the general concept that we should be getting. Okay, so we've done that. Let's go to the other one. A carrier molecule on its own may be able to prevent the HIV of cells by HIV. So it's a carrier molecule. This is quite a tricky one. A carrier molecule on its own, it can prevent the infection of HIV. So it's saying the carrier molecule on its own, without having medication, other medication, on its own, the hypothesis is, in theory, it can prevent the cells from getting infected with HIV. Why? So key word is here, prevent infected of cells. And this is again similar shape concept again coming up here. And this is about competitive inhibitor concept. This is the concept that they always use in many situations. So have a think, pause the video. What do you think? You can write down. Okay, so this is talking really about the competition. What number is this? C. So the carrier molecule, it binds to the protein on so the carrier protein, it binds the carrier molecule, it binds to the protein on the APC cell. So the antigen presenting cell, the infected cell is there and has a protein outside, right? HIV protein. The carrier molecule binds to it first. When it binds to it first, it prevents HIV from binding to it as well. Sorry, I think my understanding was a bit off here. So let me see if I can draw it out. I don't know if it's gonna work. It will not work. So not let me draw. So maybe I can. So I'm gonna try to get some drawing. Okay, still not working. Okay, so I fixed it. So let's say this is my HIV, and HIV, and I know that, and the drawing has gone. It's not good, is it? So I want to put my HIV. This is my HIV. Okay, so HIV cell. It's not a cell, is it? It's non-living. Anyway, HIV has these proteins. These proteins are outside, right? Outside the HIV. So if the carrier molecule binds to those HIV molecule, okay, if it binds to the HIV proteins, then that means that protein is no longer able to bind to the receptors of T helper cell, of human cells. So it prevents it from binding to it. So that's the question. Again, it's competition. So carrier molecule binds to the HIV proteins on the HIV itself, and so therefore the HIV will not be killed. It's there, but when it goes to T helper cell, the CD4 cell, the human cell, and it wants to bind to that human CD4 T helper cell's receptor, it goes towards it, but the protein that is meant to bind to that receptor of the cell is not there. I mean, it's there, but it's bonded with what? With carrier molecule. So it's not really free to bind to those receptor. Hope that makes sense. Okay, so let's go to question number 3. Figure this is what was showing you here. Let's see. So whenever you see a question like this, don't just skip to it like this. Don't just rush, because you might, if you understand 110%, that's fine. But if you understand like most of it but not all of it, then there's very much of a chance that somewhere here you'll get a question wrong because your understanding is poor. So these figures should be understood carefully. So figure 1 shows a part of a gene. So I'm thinking a gene, a nucleus, DNA, DNA, fine. I'm thinking, what gene is like this? Gene is double-stranded, so why is a single strand here? Oh, maybe it's unzipped. Okay, fine. It's being transcribed. Oh, it makes sense here. I have my promoter region here. Promoter region. That makes sense. It's before the gene starts. And then I have my gene here. And then what's this? Oh, RNA. I'm thinking, what's RNA? And why is it small? Oh, I'm thinking, oh, this is the mRNA, fine. And it just started, okay? Even though it's RNA, but this is our mRNA. And what's the enzyme? RNA polymerase, most likely, right? Trying to transcribe this. Okay, so once you've identified this really well, let's start cracking on. Name enzyme X. What do you think? It's going to be, let's see, enzyme X is RNA polymerase, fine, yeah? So we're doing transcription, not translation, so it will not be DNA polymerase. Estrogen is a hormone. So estrogen, so it's talking about estrogen, and I know about everything about estrogen. We know that it's lipid soluble, okay, and it affects transcription. We know that, right? We know that it stimulates transcription. It forms a complex with a receptor in the cytoplasm of target cells, and we know that this complex, so that this receptor is called estrogen receptor, right? So what is this receptor? From our A-level understanding, we know that this receptor is a transcription factor. What is that called? Estrogen receptor. It forms a complex. What's that complex called? Estrogen receptor complex. Anyway, so from the cytoplasm, it enters the nucleus and goes to the promoter region, targets the cell. Explain how an activated estrogen receptor. So you're thinking, what on earth is estrogen receptor? If you don't know, if you haven't revised and have not got this grounded about transcription factor, about a transcription factor called estrogen receptor, this will not make sense to you. So this receptor is not a receptor in the cell membrane, right? This is a transcription factor. So they're asking you, this activated transcription factor, when it leaves the cytoplasm, enters the nucleus, how does it influence? How does it affect the cell? Target cell means the cell that's being affected. So have a pause. What do you think the answer is? Okay, B. So we know that the receptor, can you see here, they said transcription factor. That's the actual word. Transcription factor binds, which is activated, right? So the complex binds, enters the nucleus, binds the promoter region, which stimulates RNA polymerase, and that always allows increase of transcription. So increase of transcription. As 2 marks, is it 2 marks? Estrogen only affects target cells. So target cells, I mean the cell that it wants to affect. Why does it not affect other cells in the body? So let's say estrogen affects the endometrial cell of the uterus. Why will it not affect, let's say, your heart cell again? So again, it's all about specificity, similar shape, similar things being there. So we know, easy mark in here, we can say the other cells don't have the transcription factor. Which one? Estrogen receptor. So they don't have estrogen receptor transcription factor. This is going to be our last question. Okay. Some breast tumors are stimulated to grow by estrogen. So in here it's saying, so estrogen is linked to breast tumors. Okay, so they stimulate. So I'm thinking estrogen increases cell division. Okay, so tamoxifen is used to treat these breast tumors. So this tamoxifen is a medication and is used to treat breast tumors. Fine. In the liver, the tamoxifen is broken down, is converted into something else called endoxifen. So in here, they are throwing different words here. New word, tamoxifen, endoxifen. Figure 2 shows a molecule of estrogen. This is my estrogen and endoxifen. So what I do here, I get this information, I simplify it. You have to simplify information. So breast tumors, okay, fine, and saying that estrogen is bad, is linked, it causes cancer, it stimulates it, but they have this medication, tamoxifen. Tamoxifen is not that important. The main important is endoxifen. This is the endoxifen, that's the active format. That's why they have drawn this molecule here, endoxifen. Okay. Now they want you to look at these two of these structures. These structures look a bit similar to each other, not 100%, but similar to each other. So figure 2, use figure 2 to suggest. So suggest means what? To give scientific reasons behind it. Does not have to be fully accurate, but just sort of give ideas. Suggest. Why do you think this medication will reduce the action of estrogen, will reduce the growth rate of breast tumors? Again, it's the same concept, similar shape, competitive inhibitor concept. Have a think, and this is our answer. Okay, so endoxifen has a similar shape to estrogen. So therefore, what happens? Endoxifen binds to receptor. Which receptor am I talking about? It's not a receptor really only. It binds to the transcription factor, okay, called estrogen receptor. So this prevents that transcription factor from being activated. So it prevents estrogen hormone from binding to it. So the receptor, what receptor is it? The transcription factor is not activated. So therefore, if it's not activated, it will not attach, going into the nucleus, attach to the promoter region. If it does not attach to the promoter region, it will not activate or increase DNA transcription.